Initiation begins when RNA polymerase binds to the promoter, a place in the DNA sequence where RNA polymerase attaches and initiates transcription. RNA polymerase then unwinds the DNA and begins mRNA synthesis. Next, in elongation, RNA polymerase moves downstream, which means it moves away from the promoter, and continues to unwind the DNA and elongate the mRNA transcript in the 5' to 3' direction. As the mRNA molecule grows longer and longer, the DNA double helix reforms. Finally, in termination, the mRNA transcript is released and polymerase detaches from the DNA. Initiation of translation binds together the mRNA and the tRNA. First, the small ribosomal subunit binds to the mRNA and specific initiator tRNA. The tRNA scans downstream the mRNA until the start codon AUG is found. A codon is a triplet of nucleotides. The anticodon on tRNA hydrogen bonds to the AUG start codon and establishes the codon reading frame. The reading frame allows the nucleotide triplets to be read in the correct groupings during translation and display the intended message. This causes the right amino acid chain to form. In the final step of initiation, the large ribosomal subunit binds to the small ribosomal subunit, and the final complex is formed. All the components are brought together through endergonic reactions with GTP molecules. Next, in elongation, amino acids are added one by one to the previous amino acid. First, an anticodon of tRNA base pairs with the complementary mRNA codon in the A site. GTP molecules are used as energy to drive this codon recognition. Next, rRNA molecules located in the large ribosomal subunit catalyze formation of peptide bonds between the new amino acid in the A site and the amino acid of the polypeptide chain in the P site. This removes the bond between the tRNA and the amino acid in the P site and attaches it to the amino acid of the tRNA on the A site. The ribosome then translocates the tRNA in the A site to the P site. At the same time, the empty tRNA in the P site moves to the E site where it is released. The mRNA then moves on to the next codon, and the process is repeated until the full polypeptide chain is formed. Termination begins when the stop codon in mRNA reaches the A site of the ribosome. A stop codon does not code for an amino acid, but rather signals when protein synthesis comes to an end. When the stop codon reaches the A site, a protein release factor binds directly to the stop codon. This causes a water molecule to be added to the end of the polypeptide chain, and the reaction breaks the bond between the polypeptide and the tRNA in the P site. The polypeptide chain is then released through an exit tunnel in the ribosome's large subunit. The tRNA then exits through the E site. Finally, the complex disassembles as the large ribosomal subunit detaches from the small ribosomal subunit.